Okay, we're going to start on chapter 11, the eye and vision tests. Looking at the objectives, you're going to look at the gross structure of the eye, table 10.4 and figure 10.27 shows you the gross structures of the eye. We're going to talk about the pupillary reflex. Pupillary reflex is a response mediated by the autonomic nervous system. It causes the pupils to constrict or dilate in response to increased or decreased light, respectively. The response can be observed in a darkened room using a small focused light. Again, the pupillary reflex controls the diameter of the pupil in response to the intensity of the light. Under normal conditions, both pupils will respond simultaneously, identically to a stimulus, no matter which eyeball gets the light. Light entering one produces a constriction of the other, a direct response as well as constriction of the pupil in the unstimulated eye. This is called a consensual response. Comparing the two responses would help you decide if you've got some sort of neurological dysfunction. For example, a direct response in the right pupil without it in the left might suggest a motor connection to the left problem to the left pupil. Lack of response to light stimulation of the right eye. If both eyes normally respond, stimulation may indicate left eye sensory input problem, or a sensory input problem from the right eye. So that's pupillary reflex. Then we're going to go on, you're going to look at the visual acuity tests. For that you use a Snelling chart. I don't know if I have anything here. That you use the Snelling chart. And that's for sharpness of vision. This is where we get the 20/20 from. The 20/20 means that a normal person what a normal person can see at 20 feet, you can see at 20 feet. And that's the chart with the letters and the directions and all that. A person with myopia. Myopia is nearsightedness. And let's see if we've got a slide for that. We do. Here we go. Okay. Emmetropia is normal vision. And as you see here, let me find a pencil. As you see here, the light, the vision, the image is focused directly on the back of the eyeball. That's emmetropia. Now, if you're myopic, if you're nearsighted, your eyeball is too long. So as you can see where the vision is properly focused right here is a little bit ahead of the back of the eye. So by the time it gets to the back of the eye and the retina where it's picked up, it's already started to disperse, and that's what gives you the fuzzy vision that you've had. You use a concave lens to correct that. What it does is adjust the focus of the image so that it's right on the back of the eye. Now, if you've got a hyperopia or farsightedness, the image focuses behind the back of the eye. And so you would use a convex lens. Again, this is figure 10.35, should be in your book. Use a convex lens to correct that. Then astigmatism. Astigmatism is an irregular shape in the eye, in the cornea, the lens, on the surface of the cornea or the lens. The irregularities, cause a distortion. As you can see here, instead of going and being focused, they're either going straight in or they're sent off in another direction. So the irregularities cause a distortion of the visual perception, and what you'd have to do is you have an uneven or more of a cylindrical lens to correct the astigmatism. Now for the astigmatism we use a different chart. It looks like like a bicycle wheel with the spokes radiating out. When you look at that, you're going to look for spokes that are either not there or blurred or moving in another direction, and you would have the two measurements: the degree of astigmatism, meaning at 90 degrees, which would be, if you're looking at a compass, would be due east, 180 would be due south, 270 would be due west, 360 would be 0, or 360 would be north. So where is that astigmatism occurring? And then depending on the amount of astigmatism, then you would classify it that way. Okay, going on. Your myopia and hyperopia are measured in diopters, and that's 1 over the focal length, and in meters. So it would obviously, since you're measuring the distance of an eyeball, it would be could be very small. And so that's astigmatism. You're going to use an astigmatism chart. Then we're going to look at the process of accommodation and the term presbyopia. Accommodation is the ability changes to maintain what your eye changes to maintain a clear image. Accommodation is a reflex. It's generally the ciliary muscles. They contract, they change the shape of the lens of the eye. And so normally a young eye, a healthy eye, can accommodate from a distance of about 7 centimeters, down to about 7 centimeters, and then off to infinity. As you grow older, the ciliary muscle, which constricts and changes the shape of the lens, weakens, and you get a term we call presbyopia. Presbyopia, as my ophthalmologist said, if you're over 45 and you don't have reading glasses, you're kidding yourself. Now, other articles are a little more generous than that, but generally somewhere around that ciliary muscle is weakened enough, it cannot change the shape of the lens, so you have trouble. And that's why everybody goes and buys reading glasses. Or if you have contacts, you might leave yourself a little bit myopic and a little bit nearsighted, which would take care of that. Okay, going on from there, again we talked about visual acuity, accommodation, presbyopia, why it occurs. The blind spot. The blind spot is where the optic nerve comes into the back of the eyeball. There are no rods or cones there, so you have nothing to pick up the light. Generally it's at the optic disc where it comes in. So that's blind spot. Obviously that's not the same blind spot that you have in a car. It's an optical blind spot in the eye because you have no rods or cones there. You never see that. Your brain fills that in. It's kind of like in chapter 9 when we talked about receptive fields. Your brain fills in the difference. So you don't see a blind spot. Now going back to visual acuity, when you look at the emmetropia, myopia, and hyperopia, one thing that they used to do is if somebody had trouble seeing, they would have them look through a pinhole, much in the manner that we use a pinhole camera today. And what happens is if you look at the chart at 10.35, the idea is to get the image focused right on the back of the eye. If you're looking through a pinhole, you don't have the other images that are dispersing, whether you're farsighted or nearsighted. You only get the visual part of it that is being pushed directly straight to the back of the eye and requires no focusing. That's one way that in very ancient days, but people used to help accommodate for the poor eye. And it's like using a pinhole, what we would call a pinhole camera today. Okay, positive and negative after images. Negative after images are caused by the photoreceptors in the eye, both positive and negative. They're caused by the photoreceptors in the eye. when you look at an intense, or you're staring at a light, or intensely at a color or a card or any object, and then move to a blank page, for a few seconds your rods or cones are still firing. What you see, if it's a positive or a color image, you see the opposite. If you look at a color wheel, you're going to see the opposite color from that. If it's a black and white, you're going to see the opposite. If you're looking at a white light bulb, when you see the after image, it's going to be black. If you're looking at a black light bulb, the after image is going to be white. Let's see if there's anything else I want to go through on that. The after images, 2, negative and positive. Continue firing of the photoreceptors after the stimuli is removed. Okay, you're going to be looking at that. Hello? Hello? Yes. Hi. Yeah. I think we still are.